Hi there, everybody. This is the end of our lesson last class. We just have a couple more slides to go through. Where we left off was talking about the 5 different types of white blood cells that we are going to cover this semester. First of all, we divided these 5 types into granulocytes, those that have granules or little vesicles within them containing different types of chemicals, versus the ones that do not have visible granules. Okay, so the granulocytes versus agranulocytes. We talked about how the neutrophils are by far the most abundant. They are a type of granulocyte, followed by the lymphocytes, which are a type of agranulocyte. The neutrophils are generally most abundant, meaning that they have about 1,800 to 7,800 cells per microliter. In total, all of the white blood cells are usually about 11,000, right? Again, compared to the erythrocytes, which are about 4 million, 5 million per microliter. Okay, so much less abundant than the red blood cells, but surely the most abundant of the white blood cells. The neutrophils can be identified by pretty much looking at this much larger cell than the surrounding erythrocytes here. Also, they have these multi-lobed nuclei. Okay, so this entire dark purple structure here is a lobe, or is a nucleus with multiple lobes. Now, not all of the neutrophils have exactly the same looking nucleus. In fact, they may have 3 to 5 lobes depending on the individual cell. Okay, so the neutrophils are also sometimes called PMNs, so the polymorphonuclear leukocytes. Oh my gosh, what is that? Well, poly means many, morpho means shape, many shapes of the nuclear of the nucleus. Okay, and of course they're leukocytes, which means the white cells. Okay, also if we look at the cytoplasm, right? So the rest of this space here, we can see there are lots of tiny little dots, okay, and that these dots are pretty much light purple. Okay, it's actually a little bit darker than what we'll see here soon, but it is lightish purple. And the reason that this is purple is because when these histological slides are made, the cells are stained using 2 different stains just so that we can actually see the cells, right? Otherwise, they're relatively clear or white. And so the 2 different stains are a red stain and a blue stain. The red stain binds to acidic substances, and the blue stain binds to basic substances. And so the fact that we see a roughly light purple cytoplasm here means that the neutrophils have both acidic and basic substances within their cytoplasm, therefore making their cytoplasms neutral. And so this gives rise to the neutrophil part of the name. Okay. In general, when a differential white blood cell count is conducted, that is, when you count up all of the white blood cells within a particular volume of the blood, it is even more telling to figure out, relatively how many neutrophils, how many lymphocytes, and so on and so forth there are. And the reason this is so important for the diagnostic process is that if one of these types of cells is very common, and it should not be, right? So you're seeing numbers that are higher than 7,800, right? This would mean that those cells have replicated, right? There are a lot more of those cells than you would normally expect there to be. And as a result, what that is implying is that whatever the job of that cell is greatly needed at this particular point in the patient's life. So for neutrophils, these are essentially our body's like front end, first line of defense once an invading pathogen, such as a bacteria, virus, fungus, right? One of those types of substances can invade our body. So it breaks through our skin, it's in our lungs, and essentially these neutrophils are the first ones on the scene, and they're going to start killing individual pathogen cells. Okay, so if you are fighting an acute infection, chances are your neutrophil count would be increased. The next type of granulocyte to look at are the eosinophils. These are called eosin because their granules, right, their little vesicles within the cytoplasm take up the reddish stain, okay, which is actually called eosin. And so what we can see in this slide over here is that this particular cell has lots of kind of reddish, pinkish little dots, right? So those are the acidic, essentially granules within the cell, as opposed to this over here is a neutrophil. We can see the very pale purple of the cytoplasm. We can also see the multiple lobes, as opposed to, again, the eosinophil, which is pinkish, right? And this changes depending on who makes the slide, who is taking the picture, et cetera, et cetera. But right, clearly we see that this color is very different than this color over here. Another defining characteristic of the eosinophils is that generally they have a bilobed nucleus. So here's a lobe and here is a lobe, right? So very different looking than this guy over here. And so if we were to see more than about 450 cells per microliter in a blood sample, this would indicate that the patient is experiencing allergies. Okay, so allergies or parasitic infection, so some kind of invasion of a foreign organism, which the body is responding very strongly to, right? This organism has gotten into our body and it is outside of our cells, but it's still very much in our body. Okay, so that's where eosinophils would increase. The basophils, right? The basophils take up, or the basophil granules take up the basic stains, right? That is the darker blue. So what we can see over here is lots of dark blue granules, okay? Again, as opposed to the pale purple that are actually taking up both the red and the blue stains. Basophil, right, because this stain is staining basic substances, okay? Also, this type of cell has either a bi or a tri-lobed nucleus. And so you might look at this basophil here, and you might look at this one here, and you might say, oh my gosh, I can't even see the nucleus. That is completely normal. Yes, there's a little bit of a shadow of a nucleus here. There's a shadow of a bilobed nucleus here. But in general, the fact that the basic stains are dyeing those granules, right, and they're so dark, usually it's pretty difficult to see the nucleus inside, right? But when we can see it, it is indeed bi or tri-lobed. So if these basophils became more abundant, right? So if you see more than about 200 cells per microliter, which is very, very few, right? So it's very difficult to find these if you're looking for them in the lab. But essentially that would indicate that the patient is experiencing an allergic reaction, right? So these very darkly staining granules actually contain the chemicals that are associated with an allergic reaction, such as histamine, heparin, and serotonin. Okay. Moving into the agranulocytes, remember that there are two different types of agranulocytes. These cells do not have visible staining granules. The lymphocytes are the second most abundant cells, or white blood cells within the blood, at about 1,000 to 4,800 cells per microliter. You might be more familiar with the terms T cells or B cells. If you're not, that's okay. We'll go into this in great detail later. But essentially, these are the ones, the T cells and the B cells, that are responsible for your immunological memory, right? That is, it's been in the news a lot lately about the vaccine for the coronavirus, and essentially, you know, how important it is to get the vaccine, which it absolutely is. And so essentially what the vaccine is doing is introducing pieces of the virus, or actually the mRNA of the virus, so nothing active, nothing alive, just molecules that are associated with this virus. These cells, the T cells and the B cells, learn to recognize what those fragments look like, and so they remember, right? For when you are exposed to the coronavirus, inevitably, we all will be at some point. But if you already have this memory, if your T cells and your B cells already have learned what the coronavirus looks like, they can immediately and strongly launch an immune response against the actual living virus. Okay, and so these lymphocytes not only are circulating within the blood, right, essentially learning what belongs to us and what is foreign, like the coronavirus, but also they patrol your body's tissues. Okay, so they actually kind of squeeze out of your blood vessels and they are sucked into the lymphatic vessels, right? So actually a lot of the T and B cells are going to hang out for a long time in your lymph nodes, in your spleen, in your tonsils, right? And essentially there they are looking for the invading virus, the invading bacteria. And so I bring all this up because that's why these are called the lymphocytes, right? They live for a good period of time within your lymphatic structures, right? Again, your lymphatic vessels, lymph nodes, spleen, etc. Okay, and we'll talk a lot about that a lot more in a few weeks together. Okay, what do these look like in a blood smear? Well, here again, we can see all those erythrocytes, those red blood cells, and actually barely bigger than the red blood cells themselves are the lymphocytes, right? So in the past, we've seen, the neutrophils are much bigger, and the basophils are much bigger than their neighboring anucleate, no nucleus, erythrocytes. Lymphocytes are barely bigger than the red blood cells. They have the most normal looking nucleus, right? So this right here is just a single old nucleus, okay? It is, it stains kind of patchy, right? So it's not like a solid color. And so because of that, it can occasionally be a little bit difficult to differentiate between lymphocyte and basophil. Maybe you'll see that in the laboratory. But the other thing that is a kind of characteristic of these lymphocytes is that usually this nice, pretty round nucleus is kind of pushed off to one side of the cell, leaving almost a crescent moon of very faint agranulous, right, no visible granules, cytoplasm. Okay, so here we can see the crescent moon. Here we can see a smaller crescent moon, but nevertheless, it is cytoplasm. Okay, so if you were to see an increased number of lymphocytes, it means that your body is fighting something, right? So your T and B cells might be increased if you've been exposed to the coronavirus, either in the fragmented, not dangerous form like in a vaccine, or the living virus just out in the world. Finally, the last agranulocyte and the last white blood cell that we're going to talk about are the monocytes. Usually there are up to 800 cells per microliter, so again, very uncommon to find these, but in one blood smear slide, you might find a couple. Okay, so really not that common. These cells are differentiated, or recognized rather, by their nice U-shaped nucleus. Okay, so here's a U-shaped nucleus, here's a U-shaped nucleus. Remember that any photographs or actual slides that you're going to see are actually cells that have been squashed and like smeared down. And so these used to be nice round spherical structures. And when they get smushed onto the slide, sometimes that nice U shape is going to look a little bit different. So you might see this side instead of the nice U. And so I point that out right here. All right, so this used to be a U. We can kind of almost imagine that this is a U that we're looking like down on. Okay. All right, the cytoplasm is more of a grayish blue. It is really similar to the neutrophils, which we can see over here, but the neutrophils look a little bit more on the red side than the gray side. Okay, monocytes are huge in comparison, right? So they're like 3 times the size of the red blood cells. So if you see a really big cell in the slide, probably a monocyte. And so these monocytes would increase during a chronic infection, right? The reason for that, right, a long-term battling of a pathogen is that these are the cells that are made actually in the bone marrow And after the bone marrow, they start circulating in the blood for about a day, and then they spend the rest of their lives, which can be many years actually, kind of patrolling the tissues, so like squeezing in and out of the cells of your skin, of your muscles, of your fascia, and ultimately making sure that nothing is in our tissues that doesn't quite belong. And so they are so uncommon because they only spend a day in our bloodstream and then they get out of our bloodstream, and they live the rest of their lives as something called macrophages. So macro meaning big, phage or phage meaning eater. So these are big eaters. Okay? So they're called big eaters because they can eat and eat and eat and eat things that don't belong. Okay. So those are the 5 different types of white blood cell. For the lecture, you should be able to identify the appearance of these cells. So I might give you an image of the cells and you should say, yep, that's a monocyte. You should also be able to tell me what they do, right? And if I gave you some blood test results, right, of a differential white blood cell count, you should recognize that, monocytes are, 0 to 800 per microliter. If you see that there are 1,000, 2,000, 3,000 of these cells, that will indicate that the patient has a chronic infection. Okay, and I give you this nice chart here to wrap up this little mini lesson so that you can see in one place all of the different characteristics, right? So this is what ideally the cells should look like. Of course, in the real world, they're a little bit more variable than that. Here is how abundant they should be, okay? What they look like, what they do, and any other pertinent information. So thank you very much. Make sure that you watch the next videos in this playlist and take notes on the PowerPoint just as you would in a face-to-face class. All right. Thank you very much, everybody. Differs from serum in that it contains those clotting proteins. Okay. Next, let's move down to the formed elements. Remember that when whole blood is treated with an anticoagulant such as heparin or warfarin, and we centrifuge that blood down, what we get is this nice separation between the formed elements and the plasma up here. The heaviest of all the formed elements are the red blood cells. Right? So they go all the way to the bottom. We can also call this percentage of red blood cells the hematocrit. Okay. In between the plasma and the red blood cells, we have what is called the buffy coat, and this is less than 1% of your blood, and it contains both white blood cells and platelets. Now, in general, all of the formed elements are formed in the same place. Remember that all of these substances, white blood cells, red blood cells, and platelets, are all tightly regulated. We control how many of them are in the blood at any given point, and they are purposely put in the blood. All of the other things end up there, but the formed elements are literally formed for the blood and the blood alone. Okay. The process of developing all of the formed elements in general is called hematopoiesis. Okay. Hematopoiesis in an adult occurs in the bone marrow. However, if we look at this graph at the bottom, we can see that the location of most of hematopoiesis is going to change over the course of your development and over the course of your life. And so I first want to point out what's going on when you are a fetus, right? So here's conception, here's 0, and of course birth over here. Now, as we can see, the bone marrow is not the location of hematopoiesis until after the 4th month, and perhaps this makes sense. You don't have developed bones really until after this particular time. And so before you have bones that have a distinct spongy bone versus compact bone, essentially all of your formed elements are going to be created somewhere else. And even that changes. First of all, before you even have organs, the yolk sac, yes, humans have a yolk sac. We'll get into that at the end of the semester, but the yolk sac is actually responsible for making your formed elements. Okay, some of the first organs to really start to develop are the liver and the spleen. Okay, and so both of these organs are going to slowly take over the process of hematopoiesis or formed element formation. Okay, we can see that the liver and the spleen become less important for this process as the bones become more and more developed. Okay, so by the time you're born, your bones, right, your bone marrow has fully taken over this process. Now, I do want to point out that the lymph nodes are also going to be important in part of hematopoiesis for the rest of your life. Okay, we'll talk about this more in the next section of this class, but essentially white blood cells kind of hang out in your lymph nodes, and if anything that doesn't belong ends up in your lymph nodes, the white blood cells are going to undergo mitosis after mitosis after mitosis, and they're going to make so many more of themselves and ultimately dump them into the blood to launch a full-on body-wide immune response. Okay, so that's what's happening in the lymph nodes. Only certain types of white blood cells are going to replicate there. Okay, but they do have to start in the bone marrow ultimately, right? And so we'll get into that later this semester. Let's go back to the bone marrow. We can see that the bones are sustaining the body's needs for formed elements by the time you're born. But we do see that the relative importance of each of these different bones is going to shift a little bit over the course of your life. When you are actively growing, okay, so as a child through adolescence, you are growing a lot, and as a result, you need a lot of blood, right? You need a lot of energy transported around your body, and so you are continually making more and more and more of these blood cells. And so during your childhood, your tibia and your femur, right, so your leg bones, are super important in this process. Okay, however, as your growth slows down, hematopoiesis is similarly going to slow down in these bones. Okay, so when you are an adult into old age, the primary locations of hematopoiesis are in your vertebrae, in your sternum, in your ribs, and of course in your pelvis. And of course, if you need a bone marrow transplant or donating bone marrow, generally they take bone marrow from your hips, right? And the reason for that is that the sternum is super close to all sorts of organs that we desperately need. If the physician makes a mistake in extracting the bone marrow, essentially it can get pretty bad pretty fast, right? If you accidentally poke your heart instead. And so the pelvis is relatively isolated. There's a lot of muscle around these bones. And so if there are any mistakes, it's not a lethal mistake. Okay, and just before we move on to the next slide, I want to remind you that bone marrow is housed within the spongy bone of any of the bones that we just talked about, right? So this one happens to be the femur, and so in the epiphyses we see lots of this like spongy stuff. That's the spongy bone, and within the gaps between the trabeculae, that is where the bone marrow is located, and so that is where hematopoiesis is occurring. Okay, this chart is something that we'll refer back to a couple times, but I want to point it out now because all of these different formed elements, okay, so the red blood cells, the platelets, and all of the lovely different types of white blood cells all derive from the same type of stem cell. Okay, so this is kind of the parent cell of all of these different formed elements. We call them hemocytoblasts, right? Hemoblood, cytocell, blast essentially means like a building cell, and so these are really like the building blocks of all of these formed elements. We can also call them hematopoietic stem cells, and so these are multipotent stem cells, which essentially means that they have not differentiated very far yet. That is, they can ultimately divide and become any of the formed elements that we see down here. Okay, so they are multipotent. They can become multiple things ultimately. Right, every time we see one of these divisions, what that's indicating is that a stem cell has undergone mitosis, right? And the daughter cells, or one of the daughter cells, differentiates, right? That is, it turns on certain genes, it turns off certain genes, and this is an irreversible process, which means that these cells, right, cells that grow up to become myeloid progenitor cells, can only ultimately produce the thrombocytes, the macrophages, the dendritic cells, right, and all of these guys. They cannot produce lymphocytes. They cannot produce natural killer cells. Okay, so once one of these like branching events has taken place, that indicates that the cells farther on down the line are all going to be derived from this cell, right? But these cells and all of their offspring ultimately cannot become any of those cells over there. Okay, and so it's just different levels of what is called differentiation. You cannot turn back the clock and, you know, become one of these hemocytoblasts again. Okay. Ultimately, what kind of pushes this differentiation, right, into a myeloid versus lymphoid progenitor cell are hormones, right? So depending on what you need, right, if you need more red blood cells, you'll have one hormone controlling the process. If you need more T lymphocytes because you are, you know, trying to fight an infection, you will have different hormones or growth factors, right? And ultimately, this will drive the developmental process in either direction. Okay. Once again, blast in general means building up. So the B and the B, the blast is the building cells. These are the building blocks, or you know, in other cases, they build bone or they build fibers, etc. Okay, so let us look back just a little bit to where this is occurring. Remember that hematopoiesis, right? All of those different cells are born in the same spot from those hemocytoblasts, those multipotent stem cells. Okay, and all this is occurring within the spongy bone here. After differentiation into, you know, an erythrocyte, into a monocyte, whatever the cells are going to go up to be, after that, these cells, of course, have to end up in the blood somehow. And so they're going to enter the blood through these really special types of blood vessels. These blood vessels are called sinusoid or discontinuous capillaries. And actually, we'll talk more about the different types of blood vessels in our next lesson. But for now, I want you to see that normally your blood vessels are pretty darn solid. Formed elements are, for the most part, supposed to stay inside your blood vessels, right? It only works if it's inside your blood vessels. And so, in order for these larger formed elements to actually cross over a blood vessel wall, we need super porous, super leaky almost blood vessels. And see, these blood vessels are called sinusoid capillaries. We can see that the cells lining these capillaries actually have big old gaps in between them. And so these gaps are big enough, as we can see over here in the histo image. These cells are actually big enough to cross through those big gaps and enter into the bloodstream. Okay. All right, so once again, I do want to remind you that all of the formed elements ultimately are born in this way. They are ultimately originating in the bone marrow. They enter into the bloodstream via these sinusoid capillaries. However, some formed elements, namely the white blood cells, have to continue the developmental process somewhere else. And again, that's something we'll get into when we talk about the immune system in a few weeks. Okay, so let's start focusing in on hematopoiesis by following the development of thrombocytes, that is, following the development of these platelets. Remember that all of these formed elements are derived from these stem cells, right? Some hormone growth factor is going to cause the daughter cells of these stem cells to differentiate into a myeloid progenitor cell. Okay, now these myeloid cells can still become all sorts of different things. Okay, so they're going to undergo mitosis after mitosis after mitosis, and some of those daughter cells are ultimately going to become megakaryocytes. Okay, and so what is actually going to push the daughter myeloid progenitor cells to become megakaryocytes is a chemical called TPO, or thrombopoietin. Okay, once again, this chemical, which is produced mostly by the liver and to some extent by the kidney and the bone marrow, so a lot of different organs can actually produce TPO, but essentially when it binds to receptors on the surface of a myeloid cell, it is going to trigger that cell to produce megakaryocytes. Okay, and so really what are megakaryocytes? These cells, as you can see, are like big and clunky. They have all sorts of nuclei. They have all sorts of like stuff floating around in their cytoplasm. So essentially these daughter cells are going to undergo mitosis after mitosis after mitosis. However, those daughter cells are never actually going to divide, right? So they never actually undergo cytokinesis. And so what you end up with is this big, clunky, multinucleate cell with all sorts of really cool things in the cytoplasm. Okay, this image here is showing us the bone marrow up here. So this is where those megakaryocytes are first produced. And these blood vessels are sinusoid. So this is how all of the formed elements are going to get into the blood from the bone marrow. And so the way that we go from megakaryocyte, big clunky multinucleate cell, to these tiny little platelet fragments of cells is that the megakaryocytes actually extend a piece of their cytoplasm into the vessel. So ultimately, the shear flow of the blood is actually going to break off tiny little pieces. And so these pieces of megakaryocyte happen to have inside any of the chemicals, any of the vesicles that were present within the megakaryocytes. And so platelets are essentially little pockets of all sorts of different chemicals. These chemicals, as you might imagine, are all involved in the clotting pathway. And so we'll talk about that in just a moment. Before we do, I also want to point out that these platelets contain actin and myosin, and so you might recognize these terms as being part of our skeletal muscles, and in fact our smooth muscle and cardiac muscle as well. This is what allows stuff to contract, and so platelets are going to be really important in clotting and healing tissue, and they're actually going to use actin and myosin to kind of pull the edges of your cut closer together. So kind of cool. Stay tuned to later in this lesson. Platelets constantly are being made and broken down. Now keep in mind that these are anucleate, there's no nucleus in them, and so if they get damaged in any way, which inevitably that happens, there's no way to fix themselves. And so these platelets actually break down after a week to a week and a half. And so megakaryocytes are always made, and those megakaryocytes are always making more platelets. Okay. And so at this point, we have enough information to talk about how we stop the loss of blood. Okay, this process, so the clotting process itself, is called hemostasis. And I know that this sounds like homeostasis, and yes, hemostasis is required to maintain homeostasis, but please make sure that you get these terms straight, right? Hemo means blood, stasis means stop, and so hemostasis means the process of stopping the blood. Okay. Generally, this process immediately stops the loss of blood when you damage your tissue, and it's actually going to start the process of fixing your tissue right away. All right, so we'll talk about that in just a moment. For the most part, textbooks will divide this process into 3 different phases, but keep in mind that it's not like step 1 happens, that finishes, step 2 happens, that finishes, step 3 happens. Instead, all of these things are kind of overlapping and kind of feeding off of each other. So for simplicity, we are going to break it down into 3 steps, but again, keep in mind it's a little bit more complicated than that. Okay? In order to introduce the process, at this point, let's watch a short animation. Keep in mind that there are a lot more words in this animation than you actually need to know. So we're going to watch the animation, and then we're going to come back together, and I'm going to walk you through what you need to know. Okay, so the video itself didn't break down hemostasis into these 3 phases, but let's add them on because this is what every single textbook is going to tell you about hemostasis, or the stopping of blood. The very first thing that happens is what is called a vascular spasm. Okay, so this is the phase that starts seconds after the injury and lasts for about a half an hour after the injury. Essentially what's happening here is that when the tissue is cut, what's going to occur is that first of all, those endothelial cells, those lining cells of your blood vessel, are going to release chemicals that are essentially signals that say, oh my gosh, like, we have a problem here, we need to fix it immediately. And so one of the primary chemicals here is called endothelin. Notice that this chemical is in blue and underlined, indicating that this is one of those words that you need to know, right? You don't need to know von Brillavon's vector and all that other kind of stuff, but endothelin is one that you should be familiar with. And so essentially, endothelin, as well as some nervous system feedback, causes the smooth muscle lining your blood vessels to contract. And so this leads to something called vasoconstriction. The wide blood vessel, previously wide blood vessel, is going to constrict, right? So the diameter gets smaller, and ultimately this allows less blood to the site of the injury, and therefore we lose less blood to the injury. Okay. Also, this chemical is going to begin signaling the remaining blood vessel wall cells, those endothelial cells, to start the process of mitosis, right? So when you cut yourself, there's actually like a hole between those cells, and so we need to make more cells as quickly as we possibly can to literally fill in that hole. What's also happening is that when this tissue is damaged, right, it exposes collagen. Right, so collagen is just naturally part of the extracellular matrix, and so when the tissue is disturbed in such a way, the collagen becomes disorganized, and we can actually see that in this diagram that I drew over. The collagen fibers start sticking into the lumen of the blood vessel, and as we talked about earlier in this PowerPoint, platelets will automatically stick to stuff unless their environment is perfectly smooth. And so this exposed collagen is automatically going to attract the platelets. And so as soon as the platelets start to attach to the collagen, they're also going to start attaching to each other. Okay, and so this is going to begin hemostasis phase 2. Now the vascular spasm is still happening, but now we are beginning the platelet plug phase simultaneously. And so this is one of the great examples in our body of positive feedback. This is an emergency situation. We need to plug the hole, stop the blood from leaving our blood vessels. And so as soon as some of these platelets stick to the exposed collagen or to each other, they are going to start releasing their own chemicals. Remember that platelets are chock full of whatever was in the megakaryocytes. Here we get to actually see what was in those megakaryocytes. Okay, so they're going to release ADP, as the video said. Okay, ADP automatically stimulates more platelets to arrive at the scene and to start sticking to each other. Okay, and again, once a platelet sticks to something, it releases more ADP. More ADP causes more platelets to come over and stick to each other, and so ultimately, this is going to intensify the process quite significantly, also quite rapidly. Okay. What's also happening here is that the platelets are going to release platelet factors. Okay, so these play a role in clotting. They are going to help to stimulate that clotting cascade. The platelets aren't just there to, kind of slap a band-aid on the problem right this second. They're also there to kind of begin the process of healing right away within minutes of the wound occurring. And so they're going to release PDGF, platelet-derived growth factor, and so this is going to bind to the neighboring cells, and it is going to stimulate the process of mitosis. Again, we need to fill in that hole that was created by the damage. Finally, platelets are going to release calcium ions. Remember that calcium causes stuff to bind to other stuff, so the more calcium there is in the environment, this is going to speed up platelet aggregation, or platelets kind of sticking together. Also, calcium is really important in muscle contraction. So remember that these platelets contain actin and myosin. So with calcium around, what's going to happen is that you're going to see the edges of the wound starting to be pulled together. Okay, so calcium is really important in that process. And so before we move on to the next slide, I do want to point out that we've all kind of seen this platelet plug before, I'm sure. So when you cut yourself between the time when blood is pouring forth from the wound and you actually develop a scab, there's that like little gummy thing in the wound. If you wash the wound after that, this thing just like pops right out and you start bleeding again. So you actually have to wait for that gummy thing to actually turn into a scab, right? Y'all know what I'm talking about. That gummy part is a platelet plug, and so it is not a scab, it is not a clot just yet, because it is still soluble in water. It can still be washed away or, bumped out of the wound. But it does set up the framework for the next step. And so the video called this next step secondary hemostasis. This is actually the process of forming a legit clot. Right? This is also called coagulation. Right? So forming of a blood clot, not just platelets sticking together, not red blood cells sticking together. We'll talk about that at another time. This is true coagulation. And so this process starts about 30 seconds after the wound has occurred. So again, this isn't step 1, step 2, step 3. This is all kind of happening simultaneously. But in order for the coagulation phase to proceed, we need the vascular spasm and the platelet plug to begin. Okay, so the goal of the coagulation phase is essentially to solidify that platelet plug, right? We want a true scab. We don't want the platelet plug to be the permanent solution to this injury. And so the coagulation phase includes the clotting cascade, right? As soon as the platelets start releasing these chemicals, it turns on all of those clotting factors. One turns on another, turns on another, turns on another. Okay. At the end of that big convoluted clotting cascade that you are by all means not responsible for learning, ultimately, at the very end, we get the conversion of what is called prothrombin, right? One of those liver proteins circulating inactive in the blood, and it is then turned into thrombin. Okay, so this is an active protein, again, turned on by the clotting cascade. Okay, and just as a reminder, this is what that clotting cascade looks like. Again, you do not have to memorize this, and you don't even have to differentiate between the intrinsic and the extrinsic pathways. Okay, so ultimately, the thrombin protein happens to be an enzyme, and this enzyme converts yet another one of these fancy proteins, fibrinogen, into fibrin, and fibrin really is the key here. Fibrin is what forms the mesh, forms the network to actually strengthen the platelet plug, right? So it actually like secures red blood cells, white blood cells, platelets, all of these different things that we're trying to get out of the blood vessel. It secures them together and actually forms something that's hard and something that is insoluble. So even when you wash your wound later, the scab doesn't just pop out. Okay, and so at this time, we also see that the platelets, which of course are surrounded by calcium that they themselves have released, we see that the platelets with their actinomycin start to pull the edges of the wound and the clot within it closer and closer together. So this really is the beginning of the healing process as we are getting rid of that clot. Of course, we don't want to keep a scab for the rest of our lives, right? Our bodies will be just covered in scabs from the different injuries over the course of time. And so what we get when a scab is formed is the beginning of clot retraction, so breaking down that clot as well. And so we know that the edges of the tissue and the vessels are pulled together by the platelets. We know that there is a lot of mitosis occurring in the surrounding cells, the ones that are still healthy, the ones that are stimulated to replicate by platelet-derived growth factor. But in order for true healing to occur, we need to get rid of this scab in the first place. And so during the repair process, the clot is literally dissolved, and this process is called fibrinolysis. So it's literally lysis, or like the exploding, the breaking down of fibrin, right? Fibrin is what actually makes the clot, and it's what we need to get rid of the clot. And so actually kind of a wild process here. Plasminogen is activated into the, or sorry, let me rewind here for a second. Plasminogen is yet another one of these inactive circulating proteins within the plasma. Plasminogen is activated into the active protein called plasma by thrombin, right? So thrombin isn't just making the clot, right, making fibrin, it is also facilitating the breakdown process as well. So plasmin's job is to go in here and start cutting apart this fibrin and breaking down that clot, right? So again, thrombin, right? Thrombo means clot. Thrombin is both pro-clot and anti-clot, right? And so plasmin is breaking down the clot while mitosis is happening in the healthy cells, and the edges of the wound are being pulled back together. Okay, so in general, these processes, hemostasis and fibrinolysis, are a good thing, right? We need to stop blood from leaving our body when we get a wound, and also we need to get rid of clots sometimes as well. And so usually we have this balance of hemostasis and fibrinolysis, and as long as this balance is maintained, we have homeostasis, right? This healthy set of internal conditions. And so this here is representing a balance, right? And surely you have seen these before in textbooks. When the balance is flat, that means homeostasis is maintained. But what happens when there's too much clotting, right? So when hemostasis outweighs the fibrinolysis, this can become a clinical issue in a lot of different ways. For example, a deep vein thrombosis, or a DVT, is essentially too much clotting in the veins of your extremities. And so this can happen well, it happens in a variety of ways, but in general, if you are not moving your muscles very much, essentially your low pressure venous return back to your heart is going to essentially allow blood to pool around your valves, right? And we're going to get into this more as we go along here throughout this unit in this class, but in general, without moving your muscles, essentially your muscles aren't helping the blood to return to your heart, so it's just kind of like hanging out in your extremities. When it hangs out too long, platelets automatically start sticking together, and as a result, they can start to form clots, right, in these extremities. And so this is a problem, of course, for circulation in your extremities, but also sometimes these clots can break free, or fragments of these clots can break free and start circulating in your blood. And so these can end up in all sorts of dangerous places, particularly when your blood vessels start to get narrower and narrower to go into smaller vessels like those in your brain, for example, like those feeding your heart muscle, for example. And essentially when these clot fragments get to these smaller vessels, they can become lodged in those vessels, and ultimately blood flow is reduced or even stopped entirely. And so that's a problem, because anything kind of downstream of that clot is not going to be fed with blood, and therefore the tissue can die. And so this is called an ischemic stroke in this particular example. Similar concept, right? A dislodged blood clot can get trapped in all sorts of different smaller and smaller vessels, including in your lungs as well. And so when a clot blocks blood flow in your lungs, it is called a pulmonary embolism, right? And so of course, all of the oxygen that you are taking into this entire half a lung is not actually getting into your blood, and so you are going to not have enough oxygen to feed the rest of the tissues in your body. Okay, what about the other way, right? What happens if there's too little clotting, right? Again, so the balance going the opposite direction. There's too little clotting, essentially you can't stop the blood flow, right? And so an example of this, as we can see it, you know, in all of these different photos here, is hemophilia. And so hemophilia is most commonly a genetic condition. It is the most commonly known is a sex-linked disorder. That is, there's a mutation for the clotting factor 8 gene that we can see right here, specifically on the X chromosome, and so this has a lot of implications. First of all, without clotting factor 8, we don't get clotting factor number 10, which means that we don't get prothrombin, and without prothrombin, we don't get fibrin, and without fibrin, we don't get a clot, right? So anytime you cut yourself, right, a hemophiliac cuts themselves, they just can't stop the bleeding, right? Because you can never really get past the platelet plug phase. Okay, so that's the first implication. The second implication is that this is on the X chromosome, which means that if you are a male and you inherit this particular mutation, ultimately that's the only one you get, and so you are stuck with this mutated, inefficient, or completely defective clotting factor 8. If you are a female, you of course get 2 of these, two alleles for this particular clotting factor, and so if one of those alleles is good, then you can still clot your blood, right? You'll just be a carrier for that particular condition. Okay, and so this disease was actually kind of passed around a lot in the European royal families a couple hundred years ago, because there was a lot of inbreeding, and so a lot of males in particular died of hemophilia because, of course, they get the hemophilia gene. The females, of course, got 2 different alleles, and even if one was a hemophilia allele, they could still use the healthy dominant allele. Okay, the final topic I want to very briefly mention for you today is leukapheresis. So this is the process of making white blood cells. Now, white blood cells, as you see in lab, have nuclei, in fact, very variable nuclei. And so because of these nuclei, white blood cells can fix the damage they accrue by circulating throughout the blood and throughout our tissues. As a result, white blood cells can live for days to decades. And so this process isn't, kind of constant, just like making red blood cells and platelets is going to be. Instead, the creation of white blood cells is a lot more tied to what you are experiencing in terms of invading pathogens. That is, anytime your body is invaded by a bacteria, a virus, a fungus, funky proteins, like whatever doesn't quite look right, it doesn't quite look like what our body should. There are specialized cells that notice that this is a problem, right? And ultimately, they release chemicals, right? And each pathogen has a different type of chemical associated with it. Essentially, these messages are specific to what is going on with your body. In general, all of these chemicals are within a class called cytokines. Okay, more specifically, the interleukins. Right? So the interleukins are a type of cytokine that can actually determine which specific white blood cells are going to be created, right? For this, we need neutrophils. For that, we need, this type of B lymphocyte, etc. Okay, and so the cells that are on the scene, and even some that are within the bone marrow and within the endothelium, for example, they release different types of interleukins. In this example, interleukin 8 is released, and that's going to do a couple things. First of all, it can actually attract certain white blood cells to the scene of infection. Right? White blood cells are kind of cool like that. They can actually wiggle out of your blood vessels and actually attack invading pathogens. And secondly, these interleukins can actually stimulate the production of very particular white blood cells. So here we can see they stimulate leukocytic progenitor cells to actually proliferate, so undergo mitosis and differentiate into exactly what we need for this specific type of pathogen. What we can see here again is that that chart of the development of all of the different formed elements. And the reason I bring it up now is to show you that if there's any mutation in this leukapheresis process, so the creation of white blood cell process, this can lead to leukemia. And remember that emia means in the blood, leuc means white, so too many white blood cells in the blood. And so there are many different types of leukemia. We're not really going to get into the details of this, but I do want to point out that the words, like the classifications of leukemias, can be tied back to this chart, right? So there can be lymphocytic leukemia that essentially indicates that there is a mutation in one of these lymphoid progenitor cells. And so ultimately all of the cells downstream, right, the T lymphocytes, the B lymphocytes, etc., are all going to contain these mutations. Okay. Myelocytic leukemia can be a mutation of the myeloid progenitor cell, or there can actually be mutations, farther on down the line. Essentially, acute leukemia is very fatal very quickly, and so this generally involves mutations in cells farther up the line, right? So in a myeloblast cell or even within the myeloid progenitor cells, essentially a mutation in these cells that are kind of farther up the stem cell line means that those mutations are going to be in more and more and more cells, and it's more difficult for your body to compensate for those mutations. And so generally that's seen in children, and the children often pass away because of these issues. Chronic leukemia is more present or more prevalent in older people. Generally, these are mutations in cells that are much farther down the line, right? If only the eosinophils are containing this particular mutation, then some of the other cells might be able to compensate for that problem. Last class, we ended by talking about hematopoiesis, right? So the making of all of the formed elements of the blood. We talked about thrombopoiesis, making platelets. We talked about leukopoiesis, making white blood cells, which of course we'll talk about in a lot more detail in the immune system. And now we get to erythropoiesis. So this is the creation of erythrocytes, or the red blood cells. And so erythropoiesis is kind of happening constantly within our body, but it increases in response to what is called hypoxia. And note that hypoxia is blue and underlined, so this is a term that you guys should be familiar with for upcoming assessments. And so hypoxia means insufficient oxygen, and this can be caused by all sorts of different things, right? So when you first start exercising, you don't have quite enough oxygen in your blood. When you move to high altitude, or you go on vacation in high altitude, all of a sudden there's less oxygen in the air, and so you don't have as much oxygen in your blood. Or this can be because you are losing blood, and therefore you have less oxygen than you should have within your blood because it is leaving your body. And so hypoxia, for whatever the purpose of it is detected in the kidneys, and to a lesser extent the liver, but primarily this process is controlled by the kidneys. The kidneys, as we'll learn, are a powerful endocrine organ in and of themselves, and when the kidneys filter the blood and they detect that there's not quite enough oxygen, they are going to release a hormone called EPO, or erythropoietin. And so this is the hormone that, of course, is going to circulate within the blood. And ultimately, as that blood arrives at the bones, right, in the bone marrow, it is going to bind to receptors on those stem cells, and it's going to stimulate those stem cells, right, the hemocytoblasts, to proliferate, so to undergo mitosis, so making more and more and more of these cells, and to ultimately, for those daughter cells to start differentiating. So that is, to start expressing red blood cell genes as opposed to megakaryocyte genes, as opposed to white blood cell genes. Okay, and so within the next day or two, the red blood cell content, or kind of the precursors to red blood cells, are going to start increasing in the blood, right? So it only takes about a day or two after EPO is released to a certain extent to actually increase the content of red blood cells. Okay, and so this diagram here gives you actually quite a few different steps in the process of erythropoiesis. I am going to focus on just a few of them. You don't have to know every single one of these terms. Okay, so first of all, this entire process is happening within the bone marrow. Remember that that location shifts a little bit over the course of your life, but in general, as an adult, your bone marrow is where this happens. All red blood cells, erythrocytes, are ultimately derived from multipotent hematopoietic stem cells, right? So these guys can become any of the formed elements. Okay, given EPO, the daughter cells of these hematopoietic stem cells are going to differentiate into myeloid progenitor cells. From there, EPO is going to further commit these daughter cells to becoming a red blood cell. And again, this differentiation process only moves forward, never backward. Once certain genes are turned on and others are turned off, there's no reversing that. So these pro-erythroblasts are really the first cells that are absolutely going to grow up to become red blood cells when they get bigger, or smaller in this case. And so these are absolutely committed to being a red blood cell. Again, this is in response to EPO released by the kidneys, and this differentiation process is tied to turning on of some genes and turning off of some other genes. Now from here, these proerythroblasts these proerythroblasts require a few different ingredients in order to ultimately become a red blood cell. Okay, a couple things that they need are vitamin B12 and folic acid. Vitamin B12 is a vitamin that you acquire through your diet, specifically eating animal products. B12 is absorbed in your intestines, into your blood, and ultimately into this process. But in order to be absorbed, it requires something from the stomach to be secreted, and that something is called intrinsic factor. We'll talk about that as we go forward, but essentially we absolutely need vitamin B12 in order to synthesize the DNA needed to make red blood cells. Okay, so B12 is required for red blood cell creation. If you don't have enough B12, that is, if you suddenly become a vegan and don't start taking supplemental vitamin B12, over time your reserve of B12 is going to go down and you won't be able to make enough red blood cells. Okay. Also, as this cell is continuing to develop, right, it's using vitamin B12 to ultimately make the DNA for these cells. At about this stage, so the polychromatic erythroblast, at this stage, we are actually making abundant proteins, right? So over here, we're making the DNA, okay? By this phase right here, we start transcribing and then translating different genes within that DNA, which will ultimately make the protein called hemoglobin. Okay, so hemoglobin you've surely heard of before, but this is a protein which contains 4 different protein subunits. Right, so if you look in here, you can see the alpha helices, and so this is just a chain of amino acids. All of these are grouped together to form a functional hemoglobin molecule. If there is any mutation in any one of these subunit genes or a problem with the translation process in any of these proteins, you get a defective hemoglobin. And so defective hemoglobins cannot carry oxygen appropriately. Hemoglobin also, in addition to these 2 pairs of protein subunits, it also contains 4 what are called heme groups. And so we can see hemo and globin making up the hemoglobin molecule. Okay. The hemes are actually iron molecules whose sole function is to bind to oxygen. Okay, and so there are 4 irons and 4 protein subunits in each and every hemoglobin molecule. And again, these hemoglobins are just being cranked out one after the other until the entire kind of immature red blood cell is filled with hemoglobin at this particular stage. Okay. In order to actually make hemoglobin, you not only need the DNA, you not only need the amino acids to build the proteins, but also you need to have enough iron. And so when we look at blood test results, oftentimes you will see iron levels and ferritin levels. Okay, iron itself, of course, is directly incorporated into hemoglobin. And so these are the appropriate levels, 50 to 175 micrograms per deciliter. That is the healthy range. We talked about transferrin last week. Transferrin essentially transports the iron throughout the blood from one place in the body to the other, so it can be recycled into hemoglobin. Ferritin is similar but different. Ferritin's job is to actually store the iron until it is ultimately needed. And so ferritin levels, of course, vary between males and females. But again, these ranges are healthy. If iron falls below these ranges, that might inhibit your ability to create red blood cells. Okay, to go back here just a moment, EPO has stimulated the differentiation of these myeloid progenitors to becoming proerythroblasts. Proerythroblasts are going to become red blood cells. They cannot become white blood cells. They can't become platelets. Okay, it's committed at this point. Certain genes are turned on, a lot of them involving hemoglobin. And so hemoglobin is created as long as we have enough vitamin B12, enough iron, and enough, of course, amino acids to make the proteins. Okay, so that is what's happening here in the polychromatic erythroblast phase. Okay, ultimately, these cells are going to be pretty much plasma membranes chock full of hemoglobin. Okay, once there is abundant hemoglobin created inside the cell, we can pretty much get rid of all of the other organelles, right? We don't need a nucleus, we don't need a Golgi apparatus, we don't need any more ribosomes. None of those things are going to become or going to be important in the life of the red blood cell. Okay, the red blood cell's job is to transport oxygen, nothing else. And so pretty much we want to get rid of all those other organelles so that they're not taking up space. They're not making the cell really big and clunky and difficult to actually make it through things like capillaries. Okay, so once we have enough hemoglobin, we actually kick out, right, enucleation, all of the other organelles, leaving behind this sac of hemoglobin only. Okay, at this phase, we call the immature red blood cells reticulocytes. And so that's the word way down here at the bottom. Reticulocytes are immature red blood cells. And these are actually the ones that are released into the blood circulation via those sinusoid capillaries in the bone marrow. Okay, note that this word is blue and underlined. It's one that you really need to know, right? You don't need to know proerythroblasts or polychromatic, none of those things, but reticulocyte is really important to know. Okay, again, once EPO is released, within a day or two, the reticulocyte count is going to increase substantially. Okay, we can also measure the reticulocyte abundance in your blood. And so usually of all the red blood cells, 1 to 2% of them are still immature. They're still reticulocytes. If you see much higher levels of reticulocytes, that means that you're producing red blood cells more quickly than expected. Okay. Any questions about this? Okay. Okay, so back to hypoxia. Why would this process be increased? For example, insufficient hemoglobin, right? So why would you have low oxygen? Well, if you don't have enough hemoglobin, you're not going to have enough oxygen. And so your body is going to start producing more blood to compensate for that lack of oxygen. And so if you have low iron, if you have low B12, right, you're not going to have enough hemoglobin. Okay. As I briefly mentioned, high altitude, right? So when you go to the mountains on the weekend, of course, from sea level by Stockton, of course, there is less oxygen in the air. And so when you first get there, it's going to be really hard to breathe, and you might get a headache, and you might get kind of dizzy when you stand up, right? All of that is because of hypoxia, because there's not enough oxygen in your blood. And so this will actually stimulate your bone marrow to start making more and more red blood cells so that you can breathe better within a few days, right? So that's why you don't climb Pikes Peak. The day you get to Colorado, you have to wait a little while for your body to catch up to the new conditions. And this is me in the corner on the north rim of the Grand Canyon a few years ago. That's about 9,000 feet in the 9,000 feet in the air. So pretty hard to breathe, especially after coming directly from South Jersey. All right, so what these up here are representing centrifuged whole blood. Okay, remember I told you that when you remove whole blood from the body, you treat it with an anticoagulant and you spin it really quickly, we get the separation of the plasma and the red blood cells, right, or the hematocrit. Remember that on average, 45% hematocrit is normal, right? And of course, this varies between males and females, but 45% is generally accepted, okay? When you have more red blood cells than 45%, right? Again, on average, so when you have high red blood cell count. This is called polycythemia. Again, big clunky word, but essentially poly, many, cyth, cells, emia, in the blood. So you have many blood cells in the blood. And so this can be indicative of disease, and we'll talk about that more in a bit. But this is actually a completely normal response to being at high altitude, right? So within a couple days of being in Arizona, if you were to look at my blood, you would have seen that I had polycythemia. I had higher red blood cell count than you would otherwise expect. Okay. Another condition that might cause hypoxia is hemorrhage, right? So losing blood. Also donating blood, right? So you lose a liter of blood at a time. And so obviously for, the next hour, the next couple days, it's going to be a little bit difficult to exert yourself because you don't have enough red blood cells to transport the blood to your tissues. Okay. Also, anemia, right? So hypoxia can be a side effect of anemia, right? So anemia, is when we have less than the expected amount of red blood cells. And so we can see here that there is a much higher plasma content than there is red blood cell content. Okay. Also, natural decay. So remember that red blood cells do not have a nucleus. Remember, we kicked that out in order to create reticulocytes that can actually circulate within the blood. We need to keep our red blood cells super tiny so they can fit through our capillaries. But, the other side of this coin, the compromise here, is that without a nucleus, red blood cells cannot repair themselves when they inevitably acquire damage from the circulation process. And so these cells do live longer than the platelets, which only live a week to a week and a half. They only live about 4 months. So all the time, your liver, your spleen is filtering out these damaged red blood cells. They are breaking them down, and we'll talk about that in a few minutes. And ultimately, they are going to recycle a lot of those components, right? But when you remove red blood cells from the blood, of course, you don't have as many, and so you can't transport as much oxygen. And so if you're making red blood cells and breaking down red blood cells is not balanced, this can also lead to hypoxia. Okay, so here are, essentially these vials right here, but looking a little bit more realistic. Before I move on, I just want to make sure that we understand this. I understand we're not so chatty this morning, but if anyone wants to tell me which of these is polycythemia, right, the first one, second one, or third one, I would be all ears. Right, so the question is, which is polycythemia?
The first one.
Perfect, thank you so much. Great, thanks guys. Hooray. Thanks for speaking up, I know it's super weird. So polycythemia is indeed the first vial right here. So more red blood cells than you would expect. And of course, this over here is anemia, and this is quite anemic. All right, so polycythemia, normal, and anemia. Good. All right, one more. A patient with hemophilia would benefit most from a transfusion of what? All right, so you can actually give whole blood, plasma, serum, or platelets to patients. All right, so what exactly is it that hemophiliacs need? All right, do they need, Savannah, do you have an answer?
B, plasma.
Awesome, very good. So plasma, and the reason for that is that plasma contains all of those clotting proteins, okay? Serum contains no clotting proteins, plasma contains all of them. And so if a hemophiliac requires a transfusion, plasma with those clotting proteins would be ideal. Does everyone understand that? If not, you're welcome to shout out or reach out to me in office hours or in the chat that I'll check at the end. Okay. The red blood cell life cycle. Again, the red blood cells are anucleate, so they only live for about 4 months. So let's talk about what happens at the end of that 4 months. Okay, keep in mind that our circulation, our heart pumps the entire volume of blood every single minute. And so ultimately your blood is going to all different tissues within the course of an hour. And so that includes the spleen, liver, and bone marrow. And this diagram down here shows how the blood flows through the bones, flows through the liver, through the kidneys as well, and here the spleen. And so while the blood is flowing through these different organs, some of them have this filtering capability. At this point, let's focus on the spleen, liver, and bone marrow. In these structures, we have cells called macrophages, macro for big, phage or phage for eater. So these are big eaters. Their function, as actually white blood cells, is to engulf things that aren't looking quite right. And of course, this can be a bacteria, a virus, or it can be red blood cells that are a little bit worn down. Okay, so again, in the spleen, liver, and bone marrow, we have macrophages that engulf and remove from the blood worn out red blood cells. Okay, once the macrophage has the red blood cells, what can happen is that the cells start to break down within it. Ultimately, hemoglobin is broken apart. Now, it is pretty expensive to make hemoglobin, and so we try to recycle as much as physically possible. Right. First of all, we can recycle the globin. All right, remember the globin is the protein component of hemoglobin. Proteins consist of chains of amino acids, and so we can break the amino acids apart and recycle them for, more hemoglobin, or we can recycle them for other types of proteins in the body. Okay, so essentially those amino acids are dumped back into the blood, and they can be used by the body in any way it needs. Okay, a little bit more complicated than that is the recycling of the heme. Remember, the heme is the iron component. It's what actually binds to oxygen. There actually is another part of this molecule, and in the spleen, liver, and bone marrow, we can actually take the heme and break it apart. Now, the iron, which of course is acquired through our diet, right? And it's not super abundant in our diet. We really need to reuse that as much as possible. And so we essentially tack iron onto our friend transferrin, which transfers the iron back to the bone marrow, right? Where it can ultimately be used in new hemoglobin. Okay, so we're not just taking, the hemoglobin and putting it into new cells, right? None of that. We're not emptying cells and refilling them with new hemoglobin. We are literally breaking down the cell, we're breaking down the hemoglobin and recycling those components to build up new red blood cells. Okay, so transferrin takes the iron back to the bone marrow to make new hemoglobin, and the one piece of this entire process that is actually just kind of eliminated from the body, thrown out, is the heme without its iron, right? And that is the pigment bilirubin. You might have heard of that before. Bilirubin, right, as we can see here, is taken from the hemoglobin, put into the blood, and ultimately it is filtered out of the blood by the liver. Okay, and so the liver is going to secrete this bilirubin into something called bile, which we'll talk a lot more about later this semester, but essentially bile is required to essentially help you absorb lipids from your diet, right? So if you eat a hamburger, all of that fat in that meat can only be absorbed into your body if we have bile. And so bile contains bilirubin, and so that's what gives bile its yellowish green pigment. Okay, so some of the bile actually goes out in the feces because of this process. Okay, bile itself and the bilirubin component has this yellowish pigment, and so this is actually what makes the feces, for a large part, brown, right? So the yellowish tint to feces is due to bilirubin. Also, this compound, this pigment, through a process we'll talk about later this semester, can also be eliminated from the blood by the kidney, right? And so again, bilirubin has this yellowish tint to it, and so when this bilirubin derivative is released by the kidney in the urine, it also gives the urine that yellowish tint as well. Okay, so bilirubin is a pigment. It is derived from the heme component of hemoglobin. It is released by the liver and the kidney into the feces and the urine. And again, it also has a role in lipid absorption. Okay, all right, so, well, first of all, any questions about that? I know that's a lot to take in. All right, so one more question before we move along. And hopefully by the end of the week, I'll have some kind of polling system so that I can get the class's feedback about these answers as well. Okay. All right, so which of the following is false about erythrocytes? Without a nucleus, these cells have a short lifespan of about 4 months, right? That is true, right? They're anucleate, they can't repair themselves. Increasing the reticulocyte count, so the immature red blood cell count in the blood, is a normal response to anemia. This is also true, right? So if you are anemic, you have hypoxia, you need to make more red blood cells. Okay, so that is also true. Older red blood cells migrate to the liver and the spleen, where they are stripped of hemoglobin and return to the bone marrow to be refilled. This is false, right? So this would be the answer to this question. It is false about erythrocytes. Essentially, this is false because older red blood cells aren't stripped of their contents and refilled. They are completely broken down and brand new cells are made in the bone marrow. Okay, D is indeed correct as well. Bilirubin has this yellowish tint, and so it helps to make feces brown and urine yellow. Okay. Most likely, this is what Jennifer has. A physician would of course use IDA as their working diagnosis and treat her accordingly. Any questions about that? Again, IDA is super common in our society, so many of you are probably already familiar with it. Okay, so let's talk about some other forms of anemia. In general, anemia is just a reduced oxygen carrying capacity. Of course, this leads to hypoxia. Okay. IDA is one type of anemia. Vitamin B12 is another type of anemia. All right, remember that we need B12 in order to make red blood cells. So if you don't have enough B12, for example, if you are vegetarian or vegan and are not taking supplemental B12, you might not be able to make red blood cells appropriately. Another, or something that might lead to B12 anemia is pernicious anemia. So this is an autoimmune disorder usually seen in more elderly patients, and essentially their immune systems attack the part of the stomach which is responsible for making intrinsic factors. So even if these individuals are taking in a lot of B12, either through their diet or as a supplemental vitamin, they can't actually absorb it into their blood. Aplastic anemia is the destruction or inhibition of the bone marrow. Oftentimes this is a side effect of something like chemotherapy, which of course attacks any cells that are replicating really quickly, like you would see in a tumor. But of course, in your bone marrow, those hemocytoblasts are also replicating really quickly. They have to continually replace the red blood cells that are lost. And so chemo can result in anemia because essentially mitosis is inhibited throughout the body, not just in the tumor. Okay. And finally, sickle cell anemia, which is actually what one of the articles that half of you are going to read over the weekend is about. And so sickle cell anemia is a condition with a single nucleotide polymorphism. So just like you guys saw, with hemophilia in Friday's lecture. This is the mutation of one single nucleotide within the DNA, and ultimately, as we can see over here, changing changing an A to a T is ultimately going to produce a different protein, so a different amino acid sequence. What this does is it makes this different shape of hemoglobin actually makes these individuals' red blood cells go from this nice biconcave disc to kind of like a horseshoe shape, so a sickle shape, when they're in low oxygen conditions. And so normal life for sickle cell anemic patients is okay unless they physically exert themselves, right? So if they're in sports or something, or if they're like, walking up a hill to get home or whatever, that is when the cells start to sickle. And that's a problem because your capillaries are essentially about as wide as a single red blood cell. And so normal old red blood cells can slip through capillaries, no big deal. But sickle cells are of course a little bit longer because of their shape, because the same cell is kind of squished. And these actually get trapped in the capillaries. And so of course blood flow stops along these capillaries, and this can be fatal in many circumstances. And so again, this is one single nucleotide mutated that leads to this very serious issue.